thank the Academy and the organizers for inviting me and giving me this opportunity to present my research interests and our research program. So my primary interests are in viruses. With the pandemic that we just all went through, I don't think to this audience I have to explain what viruses are, but just to make sure we're all on the same page, viruses in general are very simply, they contain an RNA or a DNA genome, and the genome is covered in a protein capsid, and those are very simply called non-enveloped viruses. My primary area of interest and what I'll be majorly talking through this talk is actually on envelope viruses. And what I mean by envelope viruses is that beyond this protein coat, which actually protects the nucleic acid genome of every virus, there's actually an additional layer of a lipid membrane, which are most likely derived from the host cells when the virus comes out of the infected cell. And these lipid membrane also have proteins again on top of them, which actually interact with the environment. So my primary interest comes with envelope virus here, and there are actually a lot of different examples of envelope viruses, and I've just put up a few examples here to kind of, for examples that all of you might be familiar with. So on the very left top here, we have flaviviruses, which are dengue, Japanese encephalitis viruses, Zika virus, for example. We also have alpha viruses down here, which are the best example would be chikungunya virus that most of you must have heard of, and then the infamous coronaviruses down here. So all these are actually not simulations of any sort. They're actually direct images of these viruses under an electron microscope, and they've all been put on the same scale to kind of give you a sense of the morphology differences between them and how different they actually look. So the coronaviruses here, SARS-CoV-2, and the retroviruses such as HIV in this case, and then orthomyxoviruses, which is actually the best example would be flu viruses. And for many of these, the ones I've shown here are many of them are human pathogens. And for many of these, we don't really have effective vaccines, or we actually have vaccines, but they're not perfect. And kind of a large area of interest in trying to understand these viruses from a structural perspective, which is where my expertise lies, is to actually understand how these are organized. Because as we have gone through, when we have a viral infection and such, and if we want to understand how to develop targeted therapeutics against it or targeted vaccines against it instead of the very conventional methods, we also need to understand how these viruses are actually organized. How many proteins do they have? How do they look in a spatial three-dimensional orientation? And apart from just the simple organization of how the different proteins are arranged in a virus, it's also to understand how do they interact with the external environment. How do they bind to their receptors? How do they bind to ligands? How do they actually interact with the immune system as in antibodies? And this is another part. If you want to understand the three-dimensional way of how these different proteins and components dock together, then again, structural biology really helps you get a high-resolution insights into it. And all of these also end up giving you a better idea of how to design targeted vaccines and also targeted therapeutics. So again, pretty much all my career, I've worked with viruses, so a lot of the examples here will be more geared toward viruses. And virus structures initially were started off with protein X-ray crystallography. Protein X-ray crystallography was also really jump-started the whole structural biology era. And so the examples I've given you here is enterovirus and human rhinovirus, which is basically common cold. So initially, most virus structures were actually solved using crystallography. So you have to actually make crystals of these viruses and actually then shoot them. It also worked with individual proteins. So here there's an example of one of the capsid proteins of a virus. So if you couldn't get the whole virus to crystallize, you could actually look at different components and then look at mixing it with biochemistry and other data to actually know how that would be involved in the full virus structure. But one of the caveats of having crystallography is that if you have a membrane or any kind of lipid on a viral particle, it actually does not crystallize, because once you have a lipid component, there's always some amount, very small amounts of flexibility, which leads to heterogeneity, and it's very hard to get crystals of them. And so I would say more late 90s and pretty much early 2000s, the next technique that came into the picture was actually transmission electron microscopy, but biological transmission electron microscopy under cryogenic conditions. So in short, you would have It's most commonly known as cryo-EM. And single particle cryo-EM is one of the methods using this technique, and this actually does not involve any kind of crystallization. And so that actually allowed allowed researchers to push for actually getting full structures of actually viruses that also have a membrane on them. So the examples here again, we have chikungunya virus, dengue, and then Zika. And in all of these, there's actually a membrane component. So there is a protein shell inside, you have the membrane, and then there's also the another coat of proteins on top of it. So now today, cryo-EM can also work with individual proteins. So you can also look at specific proteins on your viruses, where if you just want to see how they interact with antibodies, you have more specific interactions you're looking at. And we have also reached very quite near atomic resolutions with it, so you can actually get a very clear idea of how different proteins are interacting in these three-dimensional structures. So far, I mean, again, we are now in 2020s, and we already have a lot of information on different viruses, how their structures are, and all of them. But one thing that we still have problems with is that most of the two techniques that I told you about still require the samples to be very pure, because you need to be able to purify them, because they all require that they are very homogeneous. And when you have that kind of scenario, it becomes there is an issue when it comes to complex virus samples. And what I mean by that is like viruses that I will show examples of, which are not necessarily all homogeneous, or in cases where if you want to know how the virus is infecting a cell probably, or how it's interacting with a cellular system, and in those cases things are not homogeneous and you have very little control on actually the system itself. And in those cases, we are still limited in how much we can know. And one of the methods that we can use for studying such complex virus systems is called cryo-electron tomography, which is the main area of interest for me. So I'm just going to go a very simple thing again, how the experimental part works, just to kind of make sure that everybody understands. But very simply, we actually have your protein or your virus samples we actually have them in solution, and you actually make a very thin layer of this on actually a carbon grid, and which actually gets plunge frozen. So what you're actually imaging under your transmission electron microscope is actually a layer of this kind of a grid, which actually has ice, and your particles are actually floating in this air-water interface there. And then you can collect the images. You have your grid, and this is again transmission electron microscopy. So you have electrons pushing through the sample, and you actually collect the images on the other side. And I'm not going to go over all the processing parts for this, just in the interest of time. But we actually have these projection images that are collected in the transmission electron microscope, which actually you can use to get back and reconstruct the three-dimensional volume from which these images were collected. And that's kind of the very simple basic of how cryo-electron microscopy and cryo-electron tomography specifically works. And so the next major parts of my talk, I would like to give examples of how we can use these methods and what can they actually tell us in terms of understanding these very microscopic and even nanoscale pathogens. So again, examples. So this is chikungunya virus electron micrograph on the left, and I hope you can appreciate that they're all pretty homogeneous. On the right, I have rubella virus, which is again, I mean, I'm sure most of us have already have the MMR vaccine. The R on the MMR is the rubella virus. And the rubella virus, if you actually purify it and put it on a microscope and look at it, they actually are not homogeneous. They actually have very different shapes and sizes. So you could get a high resolution structure of chikungunya virus more easily, but with the rubella virion, because they're all homogeneous, heterogeneous, it's very hard to average between them to actually get a high resolution structure. But using cryo-electron tomography, what you can do is, because now you're constructing each of these individual virions individually, and you're not assuming that they're equivalent to each other, you can actually look at each individual virion in a whole sample, and you're not looking at it just in 2D. You can actually reconstruct it in 3 dimensions, which means that you can actually look at the top of this, bottom of this, you can move it around and actually understand what are the different layers of this virus. And in cases where viruses have very different morphologies, you can actually compare across and see how do they form these different shapes using the same type of proteins that should be in all of them. And this was used a lot for SARS-CoV-2. So it's SARS-CoV-2 again, it has more of a heterogeneous morphology in the sense that they're all circular to a certain extent, but they're not identical. And then amount of, the spike protein that most of us might have heard of is actually arranged on the surface. So again, using tomography, because you're looking at every individual particle, you can analyze each of them separately. you can actually get a sense of how they're distributed. How many spike proteins does the one virus have, that which means how many antibodies can it interact with, or what kind of interventions you may need to have, and also what kind of conformations each of these protein has on the surface. And not just on just the surface, and again, because you're looking, by the property of how these images are collected, you can actually look at even the interior. So it gives you a full information all the way from the exterior to how the interior is aligned in these particles. So again, this is just an example of another virus in the same way, which is HIV. These are actually pseudo particles of HIV, which again are circular but not always the same. They are not exactly homogeneous or identical, but again, using tomography, you can analyze each of these virions separately. You can you can easily see how the different proteins on the surface are distributed. You can also see conformational changes in how they are tilting or how they move with respect to the membrane. And again, you can also look through the virus. You can actually understand and get a direct imaging, direct data on how every part of this virus is actually interacting with each other. And all of this builds to our knowledge of understanding how these viruses actually function, how do they organize, assemble, and also how they infect cells in the other way around, and how they have to, because all these proteins have to interact with each other to actually cause infection in cells. So far, what I've shown you are examples of actually, just purified wild-type virus and how they actually interact with each other. And in many cases, they have been characterized to very high resolutions. But one advantage of using cryo-electron tomography is that you can also look at biological processes. So the next example I'm going to show you is actually where you can use cryo-electron tomography to look at how different steps in the life cycle of viruses. So my primary example here is going to be with chikungunya virus and how it and one of the steps on how it enters. So very simply, chikungunya virus actually has a receptor on the membrane, which actually it interacts with, and then it gets engulfed into the cell. Inside the cell, it actually undergoes acidification, which causes changes in the viral coat, and that actually drives what is called a membrane fusion reaction. And what this means is that because the chikungunya virus has its own membrane, it has to fuse that membrane with the cell membrane in order to actually break apart, because it has to deliver its genome, for which the virus actually has to disintegrate. But for a long time, we actually had models for how this might occur, but we never had the technology, or we hadn't reached the point where you could actually start directly looking at it and see how does the virus accomplish this. And this is just an example of how a typical tomogram would look like. So these are collected under 300 kilovolt cryo-electron microscope. And what we're looking at is a three-dimensional volume, and we are slicing through it. So we have all these fuzzy particles, which are the virus. And you can actually look at them directly interacting with membrane mimics here, which are liposomes. And you can actually see, and then you can look at each of them. You can zoom into each of their interactions, and you can actually understand how the virus is. And this is black as high density, and for some people who are not used to seeing that kind of image, it can literally look very grainy. But you actually can see each, for example, on the right here, we have these very nice liposomes, which you can see the membrane by layers, and you can also look at the virus, and you can tell the difference between how the different conformations of the virus are as it is going through this process. Okay, sorry, for some reason the video is actually not playing, but what I want you to show is that between the different viruses that we can look at, we can actually see how the protein rearrangements are taking place. We can see how the different membrane regions here, we have the membrane of the virus and the membrane of the liposome, how do they come together. We can also look at how they actually fuse, what kind of hemifusion will take place, and how the nucleocapsid gets released into the cytoplasm, in this case, inside the liposome. We can also look at how the surface proteins now actually have moved out and what kind of conformations these proteins can make. So pretty much every step of the process, you can actually now directly image them and get a direct idea of how these structures look like and how it's actually accomplishing this fusion from a complete virus particle, fusing it and actually releasing its genome. It's kind of a pity this doesn't work because it actually puts together all the data. I actually have it, but I don't know how long it will take to. Yeah. Anyway, so moving on, along the time when I published this work on the chikungunya virus with liposomes, by that time people were also looking at these viruses in terms of the whole cell, in terms of how they infect, and because now we have the technologies to actually look at direct cells and how they interact. So the first example here is actually of chikungunya virus again, but in this case, it's actually budding. And what I mean by budding is that the virus is already infected, and you're looking at how the virus gets out of an infected cell. Actually, how does the progeny virus work? And in this case, it's actually a full cellular edge of a cell, and you have all these virus particles that are getting out. And you can actually, again, because these are three-dimensional volumes, you can actually look at how the viruses are budding. You can see where the plasma membranes are, and you can see that a lot of the ribosomes get actually moved near the area where the virus assembly and buddings are taking place. So pretty much all the components that are happening during this, now we can actually have direct data on how that works. And I think all my videos are not going to work. Yeah. Okay. So in the same way, this work was done in Cambridge, UK, which was again during the COVID pandemic. And this way they actually looked at the SARS-CoV-2 infection and directly again into cells. And this was not even the edge. This was actually at the center. So they are actually, again, because it's again three-dimensional volumes, you can segment it, and you can actually see where the transport vesicles are. You can see where the viral membranes are forming. They can actually figure out how these viral replication factories are happening, where the spike proteins are getting produced, how the virus actually buds out, and all information not from reconstituted systems, but actually directly when the virus is infecting a cell. And so with that, I actually would like to come to the conclusion of my talk, that using cryo-electron tomography and sub-tomogram averaging now, with these methods, we can now actually probe more and more into the cellular context and actually move towards what you could call structural cell biology, where you're looking at cell biology problem, but you can actually also look at them from a structural perspective without having to always look at them, trying to purify them or trying to put them in an in vitro context. Or many times where we already have structures from protein crystallography or cryo-EM, you can now look back and see how they actually fit into these larger contexts in the context of the cell, where there is a lot more, many things happening at the same time. It also allows for visual tracking of biological processes like the ones I gave you examples of. We can look at how viruses enter, how viruses exit, for example, and in many cases, though all of my examples are viruses because I'm a virologist, but at the same time, these can be applied to a lot of different, many other systems, and almost all biological systems have now been studied using these methods. And again, there are also, this is one of those fields that are actually moving very fast nowadays, and we have a lot more methodologies that are getting added on to the cryo-electron microscopy pipeline, which also makes sectioning, looking at things all the way up to the nucleus, which is the thickest part of the cell, actually possible, that we can actually get high resolution information from this. And with that, I just have a last slide showing so far what my lab at Indian Institute of Science is actually fairly new. So we have just about reached the two-year mark. So most of the data that I showed you was from my postdoc work. My current lab here, we are actually more interested in flaviviruses, which comprise most of dengue virus, Zika, Japanese encephalitis. And we are interested in using cryo-electron tomography to look at how these viruses actually enter cells and what kind of receptors do they bind to and what kind of changes do they do. And with that, I'd like to thank you all and also acknowledge the people involved in all the data that I've showed, my current lab members, and also the funding. And thank you all for your attention, and I'll be happy to answer questions.
You showed that some particles are homogeneous, some are inhomogeneous.
Yes.
Now, I understand the homogeneous, but there is a very important biology in homogeneity. So when you make an average of that for the presenting image, are you not missing some very important information?
So I think that's definitely a valid point. And so a lot of the original structures were assuming everything is homogeneous, but now I think more and more we're also considering that even things that look homogeneous can have small heterogeneity or changes or differences. And then in those cases, people have been using cryotomography and other places to actually avoid seeing that. Yes.
Your cryo-ET, can you say, distinguish between the SARS-CoV-2, its binding with the S2 and also with the TMPRSS2, the stepwise? Can you distinguish?
So technically, yes, but one thing that you have to always remember is when you're looking at it in a cellular context, there'll always be a lot of different, you have no idea what's on the cell. We still don't have much of an idea. And in those cases, we also utilize what I had very briefly at the end was like they also use a method which is called correlative light and electron microscopy, where in most cases they tag these smaller proteins, so you know where the proteins of interest are on the cell, and then you focus on those areas for your tomography data collection. And in those cases, yes, you can tell the difference, but more important is in microscopy, because everything can be seen. You need to be able to identify your protein of interest.
And I was wondering whether you can add to that the immunoelectron microscopy also. Does it help?
So so I would say they're more used complementarily. I would say that, so people actually use immunoelectron microscopy to kind of first, more of the initial ways to make sure that you have your sample correctly, prepared and all of that, and then you move forward with doing the same areas with the cryo-electron tomography. Yes.
Thank you. Nice.